In this video, we will discuss about contribution of different pathways towards cytotoxicity of CD19-CAR-NK92. So we have discussed the generation of uh, different knockouts based on the pathways that could be playing role towards this enhanced cytotoxicity of CD19-CAR-NK92 compared to the wild type NK92 cells. So the knockouts that they have prepared is this Perforin knockout, the FasL knockout, which is a dead cell receptor, and this is the lytic granule, and one is the one knockout that has both the FasL ligand and perforin. One knockout in which both this FasL ligand and perforins are not present, and they did the co-culture studies with Daudi cells and Karpas422 cells. And let us first discuss the results for this Daudi cells. So in this, they did the experiment at different effector to target ratio, starting with this 1:4, 1:1, and 4:1. And for the wild type NK92, they see a minimum cell death, which increases with increase in the effector to target ratio. And for the CD19-CAR-NK92, which is written here as reference CAR-NK92, they see a increased or very significantly increased death cells compared to the wild type NK92 for both the for all the effector to target ratios. And for this FasL ligand knockout, this is see a non-significant decrease for first to target ratios, effector to target ratios, but at for all the three effector to target ratios, they do not see a non-significant decrease, or we can say that there is no significant decrease when the FasL ligand is knocked out, meaning this FasL ligand might not be playing a significant role in the cytotoxicity. Then they knock down, then they check the results for this perforin 1 knockdown CAR-NK92 cells. In that case, the cytotoxicity decreased significantly for all the three conditions, indicating that this perforins plays a significant role in the cytotoxicity effect of the CD19 CAR-NK92. And when they check it for the dual knockout, they are able to see a significant decrease continues to be there, but there is no enhanced cytotoxicity reductions compared to the perforin knockout for 1:4 and 1:1 effector target ratio, but there is an enhanced decrease in the cytotoxicity for the effector target ratio 4:1. And they did the similar studies on the Karpas422, and here also for the effector target ratio, here also they see no significant difference for the reference CAR-NK92 and the wild type NK92 at 1:4 effector target ratio. But as they increase the effector to target ratio, they are able to see a enhanced cytotoxicity for the reference CAR-NK92. And in that case, when they check it for the FasL ligand knockout CAR-NK92, they see the similar kind of results for effector to target ratio of 1:4 and 1:1. That is the decrease in non-significant compared to the reference CAR-NK92. But for the effector to target ratio of 4:1, they see a significant decrease compared to the reference CAR-NK92. And when they check it for the perforin knockout CAR-NK92, they see significant reductions for all the three conditions. And similar to the case that we have seen for the Daudi cells, for the conditions where both FasL ligand and perforins are knocked out, the reduction in the cytotoxicity is non-significant for 1:4 effector target ratio and 1:1, and also the 4:1 target ratio. In this case, all the three effector target ratio shows a non-significant reductions in the cytotoxicity when both the target proteins are being knocked down compared to the perforin knockdown. Based on these results, they made a percentage contribution chart at different effector to target ratio, indicating what are the contributions of the different mechanisms towards the cytotoxicity effect of CD19 CAR-NK92. And they are able to see that the effect of the perforin knockout is significant, and the contributions for this contributions for this FasL ligand increases with increase in the effector to target ratio, and these things they have seen for both Daudi cells and also for the Karpas422 cells. So overall, they made a come to a point that the both mechanisms play a role to some extent. However, the role played by the perforin knockout or perforin via the release of the lactic granules is much more compared to the FasL ligand triggering apoptosis of the target cells. So I hope you understood how they validate how they check the contributions of different pathways towards cytotoxicity effect contributions of CD19-CAR-NK92. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.